I've been in communication with some of you. And I'm the current director of the Epigenetics and Chromatin Clinic. You can see the logo in the middle there. And our idea with this clinic was to build a clinic that could develop the best expertise for a limited subset of disorders. And the disorders that we are focusing on, we call the Mendelian disorders of the epigenetic machinery. And one of those disorders is KAT6A. And I'm going to tell you a little bit about epigenetics. I'm going to tell you a little bit about our therapeutic development for Kabuki syndrome, which is a related disorder. And then I'm going to talk a little bit about what we could, in the future, hopefully do for KAT6A. Let's start with something very basic. So what is DNA? So DNA is the language of life. It's how the cells know what to do. And we sometimes call the entire content of the DNA the book of life, or the human genome. And if the human genome is a book, the genes would be words. So those are the kind of words in that book that make sense. And we've had this book now for about 15 years. But we still don't know how to read it yet. And so the reason why this is very complicated is because this same text, this one book, the human genome, can be read in many, many different ways, at least 300 different ways. And the reason why I say that, there's 300 different cell types, and they all use the same book. And yet, some of these cells, like a sperm or a neuron, have very, very different components, so they must be reading the book in different ways. So how is that actually done? And one theoretical way that you can read a book in different ways is to highlight the part that you want. So theoretically, you could have a highlighting system that picks out the words that you need for a sperm, and then a different highlighter that picks out the words that you need for a neuron. But how is that actually done in cells? And I'd argue one way to think about the epigenetic machinery is to think about it as the genome's highlighting system. And so what are these? These are these, you heard a little bit about it both from Dr. Kelley and Dr. Harris. These epigenetic marks are these little tweaks to the DNA or the proteins that the DNA is wrapped around that are heritable through cell division. They're reversible, they're known to be affected by the environment or diet. And if nothing else, they add to the information content of the DNA. And there are two types. There's something called DNA methylation, and then there are the histone tail modifications. And KAT6A is a protein that affects the histone modifications. So in the histone machinery, there are four components. There are these writers. KAT6A is a writer. There's also erasers that compete with the writers. There's readers that keep track of what has been written or erased, and then there's something called remodelers that kind of move the nucleosomes around. So the way this works is that a gene is recognized by transcription factors, and there's a particular writer that's brought in. It puts on certain modifications. The writer and the eraser compete at that particular gene, and either that gene ends up being open or closed. And then you have these readers that can keep track of what has been done. So this is how, so the DNA is wrapped around these histone proteins, but the histone proteins have these tails that stick out of the nucleosome. So this is called the H3 tail, and this is called the H4 tail. And there's some very simple modifications, like histone acetylation, which is what KAT6A does, is a binary mark. You either have histone acetylation or you don't. And if you have histone acetylation, the chromatin is open and the genes are available. If you don't have histone acetylation, the chromatin is closed and the genes are not available. And this is the reason why we're so interested in KAT6A and these related disorders, because we think that the problem has to do with opening chromatin. That the genes that are needed are all there, but we just can't access them because the chromatin is closed. There's this other modification called histone methylation. It's a little bit more complicated because it has 4 states. You can have no methylation, mono, di, or trimethylation. This is an open chromatin mark, H3K for trimethylation, but then there are other histone methylation marks that are only seen in closed chromatin. So what I want you to get out of this is the histomachinery consists of writers, like KAT6A, erasers, readers, and remodelers. And there are many different histone modifications, but what I want you to know is that there are some marks that are only seen in open chromatin, like the acetylation, and then there are other combinations that are seen in closed chromatin. So what then happens when the epigenetic modifications or the machinery are disrupted? So this is the basis of our clinic. The idea with this clinic, which is myself, Jackie, and Jill work in this clinic, is that we could build expertise in these disorders and be ready for therapeutic trials, develop biomarkers, and also just build expertise so that we can be the best providers possible for the patients with these disorders. And we see a lot of patients with classic epigenetic disorders, but we also see a lot of patients that have defects either of the DNA methylation machinery or the histone machinery. And there's now about 55 of these disorders. I know this is a complicated figure, but I just want to walk you through it. That when we wrote this review, there was about 18 writers, 7 erasers, 6 readers, and 13 remodelers. And the other thing that you can see when you look at this wheel here is that almost all of them have this yellow label. So this yellow label is when the kids have intellectual disability. So basically all these Mendelian disorders of the epigenetic machinery lead to neurological dysfunction. That's a unifying feature of this entire group. There's other features. For instance, if you look at the red label, that means the kids have growth retardation, so they don't grow as well as other kids. And almost all the disorders have either growth retardation, and then there's a small subset that has overgrowth, so the kids are too big. So intellect and growth seems to be severely affected by these disorders. And then the other thing is, these disorders are enzyme disorders. So KAT6A is an enzyme. And usually you need to lose both copies of an enzyme to get a disease. But in these disorders, you only need to lose one copy to get a disease. So it's enough to lose one copy of your KAT6A and you end up with a disease. So that's another unifying feature of this entire group. They're very sensitive to the amount of the factors that are needed. And we now know a lot of these writers and erasers that are associated with the very marks that I showed you that lead to disease. And you can see, we see a lot of patients with something called Rubinstein-Tabi in our clinic. And KAT6A has overlapping function. And it's very interesting to listen to you talk and tell us about what your kids have, because we see a lot of overlap between Rubinstein-Tabi and KAT6A deficiency. Rubinstein-Tabi has similar intellectual problems. They also have very severe constipation, and they also have growth abnormalities. So there's a lot of overlap because we think that at the end of the day, maybe some of the downstream targets are shared between the different conditions. So here I'm just putting some pictures. I haven't added a picture to KAT6A because it's so new, but basically it maintains some of the acetyl marks here, just like the Rubinstein-Tabi factors. And many of the patients have some phenotypic features, but I think the other thing that's someone asked before about rare birth defects. So these conditions affect many, many genes because they regulate many, many genes. And I think that in general, the phenotype is quite broad in these disorders. There's a lot of different disease things that can go with a condition because of the fact that this is a transacting factor that interacts with a lot of other genes. So maybe depending on what variation you have in those genes, that's going to decide how your child ends up having, what feature your child ends up having. So to summarize here, the Mendelian disorders of the epigenetic machinery, like KAT6A, these are genetic disorders that disrupt the epigenetic marks. And they have they're very dosage sensitive, and that indicates that the marks are very tightly regulated. And we know now of many conditions that are caused by mutation in these writers and erasers, including Kabuki syndrome and KAT6A deficiency. So the question that we always get, and you know, the dogma in genetics has been for a long time that you cannot treat intellectual disability after birth, that all the damage has already occurred and there's nothing you can do. However, we have some evidence, and I want to show you the evidence, that there is some flexibility in these disorders to treat them after birth. We just have to figure out exactly how to tailor that treatment to the individual conditions. So, and because I have most data from this disorder, called Kabuki syndrome, I just want to show you the data so you can understand what can be done in the future for KAT6A. So Kabuki syndrome is a disorder where there are two different genes known. The kids are all small, the kids all have some intellectual disability, and that many of the kids have immune problems. And so neurologically, they have intellectual disability, and they also have very bad visual spatial defects. So what that means is their drawing is worse than a lot of other things. So they basically are very poor drawers. It also affects how they interact and learn in their home. So they often have great difficulty with kind of figuring out where they're going and so forth. And there's some overlap with KAT6A there. Kabuki often has hearing loss, which is not something that we see in KAT6A at this point. They often have congenital heart disease, which we do see in KAT6A. They do have some immune problems, endocrine problems, growth problems, skeletal and renal problems. So, the reason why I was so interested in Kabuki syndrome is that there are two causes. There's a writer and there's an eraser. So the writer that's defective in Kabuki syndrome is called KMT2D, and it puts on an open chromatin mark that helps keep the genes accessible. The second cause that was found is this eraser that removes a closed chromatin mark, and again, even though it's a different mark, they both have in common the inability of opening chromatin, just like in KAT6A. So my idea, which was a very simplistic idea, but this was our starting point, that maybe the balance between open and closed chromatin is very tightly maintained, and maybe in Kabuki and KAT6A, the balance is tilted towards closed chromatin, and if that was true, we should be able to use drugs that open chromatin and get some therapeutic effect. So to do that, I developed a mouse model for Kabuki syndrome, where we knocked out the enzymatic function of KMT2D, and we were able to show that the mice had many phenotypes that we see in the patients. They're small, they have skeletal abnormalities. This is work from Jill, who you guys can talk to later. She's been very focused on the growth in many of these disorders. And because we were interested in intellectual function, we did a wide variety of behavioral tests in these mice. So to look how they compare to other mice that don't have these mutations. And this is an assay called novel object recognition. So here you put identical objects into a cage, and then the next day you put a new object. And a normal mouse should spend more time with the new object because they remember already seeing the other object. The mice are curious. But when we did this in our Kabuki model, what we saw is that there was no difference on the first day, but on the second day, the mice they spent less time with the new object.